This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: What is CRISPR divided by Cas9? Answer: CRISPR divided by Cas9 is a genome editing tool derived from bacteria. Question: What does CRISPR divided by Cas9 consist of? Answer: CRISPR divided by Cas9 consists of a guide RNA, which is complementary to a target DNA sequence, and an endonuclease, which makes a single or double-strand break at the target site. Question: How are imperfectly cut segments repaired in CRISPR divided by Cas9? Answer: Imperfectly cut segments are repaired by nonhomologous end joining, resulting in accidental frameshift mutations, or a donor DNA sequence can be added to fill in the gap using homology-directed repair. Question: What are potential applications of CRISPR divided by Cas9? Answer: Potential applications of CRISPR divided by Cas9 include removing virulence factors from pathogens, replacing disease-causing alleles of genes with healthy variants, and specifically targeting tumor cells. Question: How does CRISPR divided by Cas9 work in genome editing? Answer: CRISPR divided by Cas9 uses a guide RNA to bind to a specific location in the genome, and the Cas9 enzyme cuts the DNA at that location. This cut can then be repaired by the cell in different ways, such as by adding or deleting specific sequences, or by replacing the entire gene. Question: What is the significance of CRISPR divided by Cas9 technology? Answer: CRISPR divided by Cas9 is a powerful tool for genetic research and has the potential to revolutionize medicine by providing new treatments for genetic diseases, cancer, and other conditions. It also allows for faster, more efficient, and precise genome editing than previous methods. Question: How has CRISPR divided by Cas9 been used in the medical field? Answer: CRISPR divided by Cas9 has been used in the medical field for a variety of purposes, such as removing virulence factors from pathogens, replacing disease-causing alleles of genes with healthy variants, and specifically targeting tumor cells. It is also being researched as a potential treatment for genetic disorders such as sickle cell disease and beta thalassemia. Question: What are the potential ethical concerns surrounding the use of CRISPR divided by Cas9? Answer: The potential ethical concerns surrounding the use of CRISPR divided by Cas9 include the possibility of creating designer babies or genetically modifying humans in ways that could be harmful or unfair, the potential for unintended consequences of genome editing, and the possibility of creating a new class of genetically modified organisms that could disrupt ecosystems. Question: What is the difference between single-strand and double-strand break in CRISPR divided by Cas9 genome editing? Answer: Single-strand break refers to a break in one strand of the DNA double helix, while double-strand break refers to a break in both strands. Double-strand breaks are more difficult for the cell to repair and can result in larger changes to the genome, such as deletion or insertion of genetic material. Single-strand breaks are easier to repair and typically result in smaller changes to the genome. Question: How is the efficiency of CRISPR divided by Cas9 genome editing measured? Answer: The efficiency of CRISPR divided by Cas9 genome editing is typically measured by the frequency of successful mutations at the target site, also called mutation rate. The mutation rate can be measured by various methods such as PCR, Sanger sequencing, or next-generation sequencing. Question: How does the specificity of CRISPR divided by Cas9 genome editing work? Answer: The specificity of CRISPR divided by Cas9 genome editing is determined by the guide RNA, which binds to a specific location in the genome. The gRNA is designed to be complementary to a specific target DNA sequence, which allows Cas9 to make a cut at that precise location. Therefore, the specificity of CRISPR divided by Cas9 genome editing is determined by the design and specificity of the gRNA. Question: How has CRISPR divided by Cas9 been used in agriculture? Answer: CRISPR divided by Cas9 has been used in agriculture for a variety of purposes such as crop improvement, disease resistance, and insect control. For example, it has been used to create crops that are more resistant to environmental stressors such as drought, to modify the genes responsible for crop yield and quality, and to develop crops that can resist specific pests or diseases.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.